Every living cell needs instructions, and nucleic acids are the molecules that store and help use those instructions. The two main types of nucleic acids are DNA and RNA. DNA stands for deoxyribonucleic acid, and it mainly stores genetic information. RNA stands for ribonucleic acid, and it helps the cell use that information, especially when the cell needs to make proteins. So in simple terms, DNA stores genetic information, while RNA helps the cell use it. We already discussed this flow in the central dogma video, so here we'll focus more on the structure of nucleic acids themselves. Nucleic acids are polymers. In simple words, they are large molecules made by joining many smaller units together. These repeating units are called nucleotides. Each nucleotide has three main parts: a phosphate group, a 5-carbon sugar, and a nitrogenous base. Now, the sugar is not exactly the same in DNA and RNA. In DNA, the sugar is called deoxyribose. In RNA, it's called ribose. They are almost the same, but deoxyribose has one less oxygen atom than ribose. That tiny difference is actually part of the reason why DNA is called deoxyribonucleic acid and RNA is called ribonucleic acid. Now let's talk about the nitrogenous bases. There are 5 main bases found in nucleic acids: adenine, guanine, cytosine, thymine, and uracil. Adenine and guanine are called purines. These are larger bases with 2 rings. Cytosine, thymine, and uracil are called pyrimidines. These are smaller bases with 1 ring. DNA contains adenine, guanine, cytosine, and thymine. RNA contains adenine, guanine, cytosine, and uracil. So the easy difference to remember is this: DNA uses thymine, while RNA uses uracil. Now, there is one small term that is useful to know. When a nitrogenous base is attached to a sugar, we call that a nucleoside. But when a phosphate group is added to that nucleoside, it becomes a nucleotide. So a nucleoside has a sugar and a base. A nucleotide has a sugar, a base, and phosphate. And nucleotides are the units that join together to form nucleic acids. When nucleotides join, they form long chains called polynucleotides. The bond that connects one nucleotide to the next is called a phosphodiester bond. A phosphodiester bond forms between the sugar of one nucleotide and the phosphate group of the next nucleotide, linking nucleotides into a chain. This joining happens through a condensation reaction. That means when the bond forms, a molecule of water is released. And the opposite can also happen. If a polynucleotide needs to be broken down into smaller units, water is added to break the phosphodiester bonds. This is called hydrolysis. So condensation joins nucleotides together, and hydrolysis breaks them apart. As many nucleotides join, they form a long chain with a repeating sugar-phosphate backbone. The sugar and phosphate groups form the backbone of the molecule, while the nitrogenous bases stick out from it. And this is the key point. The sequence of nitrogenous bases stores genetic information. The sugar-phosphate backbone is mostly repetitive, but the order of the bases can change. Different base sequences carry different instructions, just like different arrangements of letters can form different words and sentences. Now let's focus on DNA. DNA is usually made of 2 polynucleotide strands. These 2 strands twist around each other to form the famous double helix structure. You can imagine DNA like a twisted ladder. The sides of the ladder are the sugar-phosphate backbones, and the steps of the ladder are pairs of nitrogenous bases. These bases pair in a very specific way. Adenine pairs with thymine. Guanine pairs with cytosine. This is called complementary base pairing. The base pairs are held together by hydrogen bonds. Adenine and thymine form 2 hydrogen bonds. Guanine and cytosine form 3 hydrogen bonds. This specific pairing helps DNA keep its structure and also helps genetic information be copied accurately. The pairing also makes sense because of the shape of the bases. A larger purine pairs with a smaller pyrimidine. This keeps the width of the DNA molecule consistent. If 2 purines paired together, the DNA would be too wide. If 2 pyrimidines paired together, it would be too narrow. Another important feature of DNA is that the 2 strands run in opposite directions. One strand runs from the 5' end to the 3' end. The other strand runs from the 3' end to the 5' end. This arrangement is called anti-parallel orientation. So, DNA is double-stranded, has complementary base pairing, and stores genetic information in the sequence of its bases. RNA is also made of nucleotides, but it is different from DNA in a few important ways. First, RNA contains ribose sugar instead of deoxyribose. Second, RNA uses uracil instead of thymine. And third, RNA is usually single-stranded.